Right, question 6. And question 6 is the typical one where they give you a hugely complicated scenario, but actually the questions are just your syllabus, are just the material you need to know. And some of it here is core concepts as well. Some of it even goes further back. It goes into GCSE days, however much of your GCSEs have happened. Right, it gives you the scenario of a polypeptide being made, or several polypeptides being made. The idea you're getting from the DNA, you're copying that one, you've got the exons and introns, and you're doing the splicing. An amazing diagram. You've got the DNA, pre-mRNA, then your functional mRNA after splicing, combined in 3 different ways. So your exons have been combined in different ways to make 3 different polypeptides from the same original piece of DNA. Your questions go actually quite simple. It starts relatively easy. Process X and process Y. Please, whenever you see a complicated scenario, think back that actually your answers have to be to do with your syllabus. X is how you get from DNA to RNA language. So what is it that translates it? Oh, sorry, that transcribes it from DNA to mRNA. That was your RNA polymerase. That was just simply transcription. The process X is transcription. Process Y, you're getting from the RNA language into the polypeptide. Hold on a second, that's made up of amino acids, so amino acid sequence, from the mRNA language to the amino acid language, which is translation. So that is your Y sorted. Technically, that's GCSE, just embedded in a really complicated diagram. It then asks you, how is this diagram disproving the one gene, one polypeptide hypothesis? If you have one gene to make one polypeptide, hold on a second, you have got one gene. That's your one gene. That's your one gene here. And how many polypeptides does it make? It makes 3 polypeptides. Sorry, I'm recording so. It makes 3 polypeptides. Now, that clearly it's just disproving one gene, one polypeptide, not if it tells you that there are 3 being made. So here we go, look at the mark scheme, transcription, translation, and then your 3 polypeptides are made, synthesized from the same gene. Following on here, explain why a mutation in an exon. This is not a mutation in an intron. You can't say it's going to be spliced away and it's going to get lost. Exons are the coding regions. So how can a mutation in an exon still not be visible, still not affect the primary structure? These ones are the silent mutations you're getting because of the degenerative code, because you've got several codons coding for the same amino acid. So from the 64 combinations that you get between your 4 bases and then actually only coding for 20 or so amino acids. So the degenerative code or having more than one codon coding for the same amino acids. The following question is a 2-part, hold on a second, it's a 2-part thing. It's got a very basic describe how to make messenger RNA part to go with it. And then the second one, which actually goes into the scenario above. What's that scenario? You've got your muscular dystrophy. If you remember, I've actually shown you a video which had that similar sort of diagram with the arrows. I have, by the way, not looked at this paper before giving it to you. I'm not teaching you to the paper. I refuse to do that because it gives you a false impression. So seeing this question made me feel like, I've prepared you for the right thing here. So in Duchenne muscular dystrophy, you have got that one exon which has got a mutation on it, and the exons need to be, when spliced, need to be attaching to the follow-on exon to make that functional messenger RNA. It tells you the shapes of the exons in this diagram underneath here, show whether they're able to bind to one another during splicing. So if they've got the same sort of triangle shape here, the same arrow shape, they'll fit into one another. If you remove just 50, then simply visually 49, 51, that's not going to happen, isn't it? So your actual questions go back to the question. The first part, describe how functional mRNA for dystrophin, or actually for anything, describe how functional messenger RNA is produced. This means you've got the gene on the DNA, you've got DNA helicase coming along, you have got the RNA polymerase doing its thing. If you want to go as far as Ozaki fragments, I'm kind of no doubt. Sorry, I'm somewhere else. I'm doing DNA replication. Ignore me there. So you've got helicase, you've got DNA polymerase, and you've got your messenger or pre-messenger RNA being made. You then need to splice away and splice out the introns. So up to there, it literally is just from the textbook. What is the process of transcription? Look at the mark scheme. DNA copied, description of the enzymes involved, saying that introns and exons are there, removing the introns. And now comes the part that's actually specific to that scenario. So up to here, you can actually get 4 marks just by simply describing the process of how is messenger RNA made. The last mark is for interpreting that scenario. Suggest why the mutation in exon 50 results in a shorter form. That is now making you think, hold on a second, 50 is broken, 50 can't be used, something is damaged on it. 49 would like to bind onto something, can't bind onto 51 because simply the messenger RNA fragments are not working, so the exon 49 and exon 51 can't bond. So therefore we need to skip 51 as well. So 52 is the next one it can copy to again, because that's where the exon fragments will be able to bind to one another. As per this silly sentence, the shapes of the exons show whether they're able to bind to another during splicing. Look at the mark scheme to go with that one here. Okay, can only take place up to 49. The mutation is where would be copying the whole gene. So therefore, we can't copy the whole thing. Hold on a second. I've been away on the next questions already, sorry. It would literally stop at 49. I've been already on the next question, ignore me there. So if we're stopping a question at exon 49, then we're just getting a messenger RNA that is ending at 49 and therefore shorter, therefore not the whole of the dystrophin molecule will be coded for. And here are the ones that actually I was mentally already at the exciting parts here. So the molecular patch, that sort of, well, let's just not read it thing. It binds to exon 50 and therefore we'll get a shorter than normal messenger RNA. We're getting 1 to 49. Here we go. The partly functional messenger RNA gives you 1 to 49, 52 to 79, even though the patch didn't go across 51. The patch only went across 50. It binds to that bit and disappears it. So silencing RNA would be used there. Suggest why the messenger RNA produced does not contain exon 51. That is the binding thing. 49 cannot bind onto 51. Therefore, it's being just skipped because 52 is the one where it can bind to again. So splicing cannot bind to 51 because they're not in complementary shapes. They're not in the shapes where they should be. Last question for this one. Suggest why a person treated with a molecular patch to reduce the symptoms of DMD would still be able to pass on the mutation for the next generation. If you're attacking something on the messenger RNA level, it describes for you, the whole system here, it describes to you that it's doing modifications on the mRNA level, not on the DNA level, and definitely not in the germline. So unless you're attacking it in the germline on a DNA level, you're going to have it passed on to the next generation. The mark scheme expresses it that way, that it says not germline, it's only on the somatic cells, and it doesn't even change the DNA there. So it's definitely going to still be present in the gametes.